Hey everyone, welcome back to my YouTube channel, and today I am back with another video in which I will be discussing genomics. So what is genomics? Genomics is the study of an organism's entire or a portion of its genetic or epigenetic sequence information. It aims to better understand the structure and function of these sequences as well as the subsequent biological products. It looks at the molecular processes involved in health and how they interact with dietary changes and environmental factors to cause disease. The field of genomics is known for its very extensive history, ranging from the 20th century until the current century. In the year 1977, a new DNA sequencing technique was developed by Frederick Sanger in order to sequence the first complete genome, the PHI X174 virus, which opened the door to possibilities in the field of genomics. In the year 1980, Frederick Sanger, Wally Gilbert, and Burge Paul was able to win a Nobel Prize for being a pioneer of DNA sequencing methods. In the year 1983, the location of the gene causing Huntington's disease was discovered by James Gisela. In the same year, a new technique to develop DNA was developed by Dr. Kary Mullis. In the year 1985, a method to profile DNA by calculating the number of repeating DNA sequences was developed by British geneticist Alec Jeffreys. In the year 1990, the Human Genome Project was launched with a goal to sequence all 3 billion letters of human genome. In the year 1992, techniques for testing embryos in the womb for possible genetic diseases were developed. Because of this discovery, parents were able to identify whether or not their unborn child was slightly affected by cystic fibrosis or hemophilia. In the year 1995, the Haemophilus influenza genome sequence was completed, and a year later, the yeast genome Saccharomyces cerevisiae was completed. The first cloned animal, Dolly the sheep, was also born. In the year 1997, biologists John Sulston and Bob Watterson published the genome of Caenorhabditis elegans. In the year 1999, chromosome 22 was sequenced to be part of the Human Genome Project and was completed in the year 2003, and because of this, humans were confirmed to have 20,000 to 25,000 genes. In the year 2002, the first complete mammal genome sequence was completed by the International Mouse Genome Sequencing Collaboration. While being smaller than the human genome, the mouse's genome is remarkably comparable to the human genome. This year saw the start of the International HapMap project, which aims to catalog frequent variants in the human genome. In the years 2007 to 2008, DNA sequencing production increased 70 times in a single year. In 2007, thanks to a technological advancement in the field. As a result, the 1,000 Genes Project was started in 2008 with the intention of sequencing the genomes of a sizable population of 2,500 people. And in 2010, the project's findings were published. The majority of genomics history has benefited medical, forensics, agriculture, and the effective management of diseases, particularly genetic variations. As genomics opened the door for people to be able to edit, predict, and change genes, it is frequently referred to as the Holy Grail of healthcare. The advantages of genomics are accompanied by ethical concerns. Given that DNA is a passing material, whatever changes were made to the parental gene will be passed on to the progeny. This raises the concern of a lack of consent. Future generations born as a result of DNA modification technologies cannot consent. And as genomics is still a young field, it is difficult for scientists to predict its long-term impact on subsequent generations. It also brings up the moral dilemma of genetic discrimination and human experimentation, which are frequently contrasted with eugenics. The fascinating field of genomics is aligned to our respective strengths. The fact that numerous diseases were still considered deadly just a few hundred years ago, but yet because to advances in science and medicine, many lives were saved. Discovering our ancestry, curing diseases, and preserving biological habitats can all be accomplished using genomics. Although genetic technology is still far from flawless, this brings biomedical evolution one step closer. I would like to express my gratitude to Jane for being a good friend and for being a great partner for this performance task, as well as to my parents for the support and guidance they have provided throughout my life. So that ends my video for today. I hope that you have learned something about genomics, and I will see you again for the next video. Bye!